This is Charlie Rose.
Now, in all of these discussions, we're going to come back and forth to certain brain regions, and 4 regions are particularly important. The prefrontal cortex, the ventral striatum, the amygdala, and the hypothalamus. The prefrontal cortex is involved in executive function, in decision-making, and very much what we call character formation. And defects in prefrontal cortex often can lead to an increase in aggression. The ventral striatum is involved in reinforcement. It's recruited for addiction and also for certain kinds of aggression. The amygdala is the orchestrator of emotion, both positive and negative, and it influences several brain systems, in particular the hypothalamus. Hypothalamus has many functions, but we're going to focus in particular its role in aggression and in sexuality.
So, Charlie, we're basic neuroscientists in my lab, and we want to understand some of the most fundamental questions about aggression. How is aggression, which is an evolutionarily ancient behavior, you see it throughout the animal kingdom, how is that hardwired into the brain? Where is aggression in the brain? And we've studied this problem in flies and in mice, and we're particularly interested in the relationship between the parts of the brain that control aggression and those that control mating or sexual behavior, because, as you know, mating and aggression are closely related behaviors, and in nature you often find that periods of aggression are at their highest when animals are mating, and these behaviors reinforce each other, but at the same time they're mutually exclusive. So a male will direct mating towards a female of the species, aggression towards another male. So there's kind of a paradox. How can these behaviors be mutually exclusive but also reinforce each other in some way? So we've begun by trying to pinpoint the neurons that control aggressive behavior. And we've started by looking in a very evolutionarily ancient region of the brain, which Eric brought up, called the hypothalamus. So we began by trying to measure the electrical activity of cells that were active during aggression or mating in a tiny region of the hypothalamus. And we found something quite surprising, and that is within this very small region of the brain, we found a mixture of neurons, some of which were active, turned on when the animals were fighting. This is done in males. Some of which were turned on when the animals were mating with a female. And interestingly, some of these neurons were active during both fighting and mating. About 25% of them overlap. So that was a very interesting observation. It was a correlation, and we really wanted to understand the function of these neurons. And so we began by using very modern techniques now called optogenetics to activate and inhibit these neurons. And we can pinpoint this activation with a high level of accuracy directly to specific cells in the brain that are active during aggression and turn them on and turn them off with a time resolution of milliseconds. So I'm going to show you a video of what happens to a male mouse when you activate these aggression neurons in the brain. And I should say before we show the video, some of your viewers might find the image a bit disturbing, but what we're doing doesn't hurt the mouse. And these are all protocols that have been approved by our Institutional Animal Use and Care Committee and our NIH approved. So you're going to see the mouse in a cage with an inanimate object. When the light comes on, we're stimulating these aggression neurons in the mouse. So we can actually trigger the mouse to attack a rubber glove. Of course, if there were another mouse there, he would attack the other mouse as well. So we then wanted to ask, are these neurons actually necessary for normal aggression? And so mice will normally fight with each other. For example, if you introduce an intruder mouse into the cage where a male mouse lives, very shortly thereafter, the resident mouse will attack the intruder. He doesn't like somebody impinging on his territory. Right. And so we asked, if we shut these neurons off, can we actually stop a fight dead in its tracks? And as the next video will show you, it's possible to do that. So now these mice are fighting naturally. When the light comes on, we inhibit these neurons, and suddenly the fight stops. And we'll show you that again in slow motion. So you can see the mice are fighting, and suddenly the light comes on, boom, we stop the attack dead in its tracks.
Okay, I don't understand how the neurons know to respond to light.
So the way that we do this is we genetically implant in these neurons deep in the brain a protein that comes from a light-sensitive algae. And that protein makes an ion channel through the membrane of the neuron that turns the neuron on only when light activates that channel. So we can convert these neurons into light-sensitive neurons. So that shows us that these neurons are necessary and sufficient for aggression.
There are different kinds of channels you can insert, those that activate the neurons and those that shut it off, and he can do either.
So we discovered, as we were manipulating the conditions for turning these neurons on, something very surprising. And that is that you needed high-intensity stimulation to activate aggression, but low-intensity stimulation would promote mounting behavior. So the mouse would try to mate with whatever other mouse was in the cage with low-intensity stimulation. Whether it was a male mouse, a female mouse, it would try to mount. And we could actually switch the behavior of the same animal from attempted mounting to a mixture of mounting and attack to attack just by increasing the intensity of light. So that tells us that in this tiny region of the brain, there is a mixture of neurons that are controlling both the mating instinct and the fighting instinct, and perhaps that will account or may account for the tension between the sex drive and the aggressive drive.
This is extraordinary.
It's extraordinary.
Yeah.
It also sort of explains why aggression can lead to sexual aggression.
Yeah.
It really is an amazing set of findings.
And so a lot of people think aggression is more prevalent in people with mental illness, and the fact is that mental illness on its own does not increase the rate of aggression. It's more likely to occur in individuals with histories of substance use and dependence and some other factors, especially prior history of aggression. So everyone gets very concerned when mass shootings happen and those sort of things. This person must be mentally psychotic, mentally unfit. And those are very individual situations. When you look across large epidemiological studies, it just doesn't pan out. Now, there's multiple forms of aggression. There's socially sanctioned forms of aggression, such as fighting and war. There's medically related aggression, where a medical condition makes one more irritable and aggressive. But the two big ones really are going to be impulsive aggression and premeditated aggression. And impulsive aggression is aggression that's not exactly spontaneous. So what happens is people perceive a threat or a frustration, and their threshold to blow up is just very low. And so they'll just blow up and experience or display a temper tantrum or physical aggression. Premeditated aggression, which can happen in anybody, is already thought through, and they've planned it out. The people who do that are more likely to be psychopaths, although not only psychopaths. And about the number of psychopaths, maybe 1% of the population. But people who have intermittent explosive disorder, which is the disorder of impulsive aggression, is more like 3 to 5 or 6%. So that's much more the case as far as that's concerned. And those important distinctions to put forth, because we have an idea of how to treat impulsive aggression, and we're not quite sure how to treat premeditated aggression. I think Adrian will say something more about that.
So what do we see at the neurobiological level?
What we see in people with aggression is very similar to what we're hearing at this table. So Dr. Tremblay talked about 4% of boys being chronically aggressive. Well, that's about how many IEDs you have out there. It's about 4 to 5%. But we also see the same sort of things biologically. We see problems with serotonin function, where usually the serotonin function is diminished. We see evidence of heightened other neurotransmitters that will facilitate aggression. In terms of systems, we see problems in the frontal area of the brain. We see problems in the amygdala and certain other aspects of that circuit. When we present to these individuals pictures of individuals who are angry or threatening, the amygdala will overact to that stimulus compared to healthy volunteers. And that also correlates with how aggressive these people have been over the course of their lives. Now, an important thing going along with this is serotonin and amygdala really have to do with sort of the tendency to be aggressive, but what makes you aggressive in the here and now has to do with how you interpret social signals. And so people who are aggressive, and it may very well be because they've been aggressed upon as a child, tend to have problems with what's called social information processing. So they don't take in enough information about what's going on, and they make a hostile inference as to what the other person is doing. So you can have a situation where your threshold may be high or low, and where you're coming into the day, is that you're primed to think that somebody brushing up against you or looking at you funny is threatening to you or frustrating to you. Then you got an overactive amygdala. You've got a serotonin system in the frontal lobe's not working so well.
Right.
Brakes are bad. You got high accelerator. Brakes are bad. You're going to have a crash.
Now, it's interesting. When Charlie and I did a program on depression, one of the key things that emerged, of course, is the treatment of depression often is designed to increase the level of serotonin. Is there an increased incidence of this kind of aggression in people who are depressed and have low levels of serotonin?
Interestingly, not necessarily. The early studies that I did were interesting in that we saw this problem in individuals who didn't have a primary mood disorder. And what we think is going on is that their brain systems that are involved in activation are not working well. And they will make a suicide attempt when they're severely depressed because that's the arousal that's going on. I feel terrible. I want to end my life. Their low serotonin then is bad brakes, and it's
turn it against
themselves rather than someone else.
Adrian, let me talk to you about certain individuals who cannot control their impulses.
Yes, I mean, Emil has been talking about people in a hospital context. What we do is work with people really at the street level, people who are violent, psychopaths, even killers.
Can I just ask, what does psychopath mean?
Yeah, it's an individual who lacks conscience, lacks remorse, lacks guilt, and because of that lack of normal emotional feelings, there's no barriers, so they do outrageous things, and they're stimulation seekers, they are impulsive.
Yeah. There's really two parts to it. I mean, there's the cold, calculating, callous type, which is really the core of it. And then you have the antisocial lifestyle, which is, doing horrible things to other people.
And we've been studying their brains using brain imaging to look to see what part of the brain may not be working right, or what part of the brain may be physically different even. What we find in normal people, of course, is that their prefrontal cortex is working well, as you see there in the green. If we can have the next slide, this is where we brain scan murderers, and what we see here are murderers who are impulsive, they don't plan their murders, they're very hot-blooded in terms of their homicides. And what we see there on the left is poorer functioning in that very frontal region of the brain.
Right.
So why is it that that part of the brain can predispose to aggression? It's the part of the brain that's involved in checking on impulsive behavior. We all get angry at times, don't we? What stops us lashing out? We have a good frontal cortex that's working well to regulate and control our aggressive behavior. So those are hot-blooded murderers. But what about the cold-blooded murderers? When we brain scan them, lo and behold, they have pretty good frontal functioning, which in a way makes sense because these are killers who premeditate their homicides. They're planning ahead. So they've got the wherewithal to do that. So the interesting question becomes, what is it then that's producing them to be violent in a predatory fashion? So let's turn to the next slide where we'll look at another brain region, and this is the amygdala. On the left you can see where it's located in the brain. What we find when we study cold-blooded offenders, psychopaths, they have a shrinkage, a physical shrinkage in the amygdala. It's reduced in size by 18%, and on the right you can see the areas within the amygdala coloured in blue that are actually physically deformed. Now, the amygdala is very important in generating emotions, as Eric had said to begin with. If there's a shrinkage in the amygdala, that will reduce fear. What stops a lot of us breaking the laws of the land? Well, we're frightened about the punishments that we would get, if we were caught. But if you lack that fear, if you have this impairment to the amygdala that normally produces this anticipatory fear that stops us committing crime, well, you're more likely to commit offences in a cold-blooded fashion. There's more to it than that, however. There's yet another brain region called the ventral striatum. On the left, you can see where it's located in the brain. In the right, you can see that there's greater activity in psychopaths when they are anticipating rewards. So we have the idea that psychopaths are reward-driven. They want the goods. Exactly, just like an addiction, Eric.
So there's a stimulation to the brain?
The idea is in anticipating rewards, the part of the brain that gets hooked on rewards is firing up a lot more. Maybe that's why psychopaths are more likely to pursue rewards and gains that they want. They've got the drive to do that, and they don't have the emotional amygdala to hold them back in a way, to give them that anticipatory fear that would normally result.
You know, this is a spectacular series of findings we're discussing here, Charlie. Because when I was a medical student, none of these imaging techniques were available. And you had very little insight of what was going on in the living brain of people. We now have insights into the biological substratum of different kinds of aggressive syndromes. That's remarkable. We can just see, this is just the beginning, how much we've already learned, different categories, impulsive versus premeditated, different categories of premeditated.
Even affecting the size of the amygdala. So therefore the question is, can you, because of your reference to imaging, can you look at imaging and decide who's most likely to be
we are
aggressively violent?
It's a great question, and we are beginning to get some clues about who may be more likely to be violent in the future. So, for example, myself and colleagues brain scanned just males in the community. Those individuals with a smaller volume to the amygdala were more cold-blooded, so to speak. They were 4 times more likely to commit a violent act in the next 3 years. And that's prediction over and above prior history of violence, prior history of psychopathy. So it's not perfect prediction by any manner of means, but we are beginning to get added value using brain imaging to try and understand who are at risk for becoming the next generation of future offenders.
Are we seeing this kind of research used in trials, in criminal trials?
The key question here is that if a psychopath is, first of all, what's causing that amygdala shrinkage? Could be genetic, it could be how the brain develops, but we also know that trauma reduces the size of the amygdala in children. Neglect reduces the size of the amygdala. For whatever reason, I don't think psychopaths ask to be born with an amygdala that's 3 sizes too small. If that brain impairment predisposes them, raises the odds of them doing the terrible things they do, the fascinating question is, to what extent do we hold them fully responsible for their actions?
This is not DNA evidence. This is not like saying, I'm responsible, David is not. Right. Definitively. Here, this is a probabilistic statement.
Yes, of course.
And that is imperfect in front of the law.
Absolutely.
So I think as techniques get better, and we see that not only the amygdala, but when the amygdala is associated with this abnormality, that means danger. When it's associated with another abnormality, it's unrelated, then we'll be in a better position.
Human parents nurture their young, and this behavior is absolutely essential for the proper development of the child. In addition, parenting is one of the strongest and most enduring social bond in human societies. Now, remarkably, parental behavior is widely conserved in the animal kingdom. In mammals, females lactate, and therefore they take primary responsibility of the parental care. As can be seen in this very nice slide, this female chimpanzee is watching over the first step of her child. So females are very frequently maternal, and not only in mammals, but also in some species of birds, frogs, reptile, insects. Now, what about fathers? Well, the contribution of males in parenting is very variable. In some species, as seen, for example, here in this silverback mountain gorilla playing with his infants, in some species, males are paternal, they nurture their young. In some other species, males attack the children and sometimes kill them. So I'm a neurobiologist, and my group used a laboratory mouse to try to understand the basic biology of parenting behavior. We would like to identify the brain areas that are involved in driving parental behavior, and we would like to understand how these brain areas are regulated in order to have animals that are parenting and some animals that are neglecting their infants. Now, in females, mothers as well as non-mothers are spontaneously maternal, which means that when they are put in presence with pups, they will spontaneously build a nest, they will retrieve the pup to the nest, they will groom them, and they will huddle with them for long periods. In contrast, males are infanticidal. They will readily attack the pups and kill them. However, males that have access to the females become paternal 3 weeks after mating with a female, which correspond exactly to the gestation time in mice. In other words, males who become father also become paternal. So we took advantage of this extremely interesting paradigm and differences in behavior between males and females and fathers and infanticidal males to try to understand what are the brain areas involved in those behaviors. The first question we ask are, what are the neurons that drive parental behavior? And in the first set of experiments, we identify a specific set of cells in the hypothalamus that are activated during parental behavior. We then ask, are these neurons required for the parental drive? So in a subsequent experiment, we genetically ablated these neurons in parental males and parental females. And surprisingly and remarkably, now these animals neglect their infants or attack them. So this experiment suggests that these neurons are required for parental behavior. In the next experiment, we ask whether the activity of these neurons was sufficient to drive parental behavior. So this time we took aggressive males and we artificially stimulated these nurturing neurons. Amazingly, these aggressive males stop attacking the pups and instead they groom their infants. So what this experiment says is that the activity of these neurons is sufficient to drive parental care. In another experiment, we identify a set of cells in a different area of the hypothalamus that is activated when aggressive males attack their infants. So we call these the parental neglect neurons. And in another experiment, we activate these neurons in females and found that these neurons now, these females, instead of caring of their infants, now neglect or attack them. So what overall this series of experiments suggests is that the brain has two components, a set of cells in the hypothalamus that drive parental behavior and another set of cells that drive parental neglect. We are very excited by these results because it opens new opportunity to understand the control of parental behavior and possibly why some animals are parental and some animals are neglecting or attacking these infants. Now, parental behavior is widely conserved among animals, and so these also raise the possibility that the function and the regulation of these cells is widely conserved across the animal kingdom.
I have one question before we go to Suzanne. How do you stimulate the neurons to make the aggressive males more nurturing?
So we use modern methods in neuroscience called optogenetics that enable to shine light on neurons that have been genetically modified and have an ion channel that is light activated. So in other words, we drive the activity of genetically defined population of neurons.
That's fascinating. Suzanne, let me talk about how it's all evolved, parental behavior.
Okay, I think what's very nice about what Catherine has told us is that parenting behavior is conserved across animals. And it's not just that it's conserved. What's really interesting is that it also varies. So there's some similarities between species that aren't very closely related, and there's some differences between closely related species. And what our work is trying to do is to understand the evolutionary basis for some of these parenting behaviors, and why is it that you have some parenting that's very similar and some parenting that's very different. So as we can see in this video, this is a video of a gorilla mother and her infant. There are a lot of similarities in the behavior between how this mother interacts with her babies and how humans interact with their babies. And this is one of the things that we really want to understand is what is it that's similar about humans and our caregiving and our parental behavior and other animals. And the biology of of mammals, females, means that there's hormones that are very, very important in determining lactation, so driving the production of milk and driving the letdown reflex. And these hormones also seem to be very important in regulating the behavior of mothers and their infants. Two of these hormones are oxytocin and prolactin. So at the end of pregnancy, the hypothalamus, which is the same part of the brain that Catherine discovered these important parenting neurons, produces a hormone called oxytocin, which is primarily important physiologically in the production of milk and lactation. However, it also is very important in driving maternal behavior. So oxytocin has these secondary impacts on the brain, such that when oxytocin levels are raised, females bond with their babies, and this hormone is incredibly important in driving this mother-offspring relationship. Another hormone that's produced in late pregnancy is prolactin, and prolactin is also important in the production of milk, but again, it also has these knock-on consequences for maternal offspring bonding. And I think that's actually really interesting, because we have this biology that allows females to produce milk, but they also are incredibly important in driving the relationship between mothers and their offspring. And these hormones also have been shown to have a lot of, be very important in driving social bonds in general with other animals. So it seems like one of the basic relationships between two individuals for mammals is the mother-offspring bond, but also these hormones are important in determining pair bonding, so the relationships between males and females, and also in more widely social relationships. And there's been some fascinating work done by teams understanding the evolution of parenting behavior and understanding the evolution especially of pair bonding between males and females. And in voles, there's a very nice system where some vole species are polygynous and some vole species are monogamous. And the prairie vole is an excellent example of a monogamous vole species where males and females form these long-term pair bonds, and both are nurturing towards their young. And what we found in this, what has been found in this system is that not only are there higher levels of oxytocin, but in the males there's a similar hormone that's produced by the hypothalamus, vasopressin, which seems to be very important in determining both parental or paternal behavior and pair bonding behavior. And in the monogamous species, there are more vasopressin receptors and higher levels of vasopressin. So we have oxytocin in females that seems to be very important in driving pair bonds and maternal behavior, and in males we have vasopressin, which plays a similar role in driving pair bonds and
this vasopressin work was done by Tom Insel.
Yes, it was done by Tom Insel and Young, and they did a fascinating series of experiments determining both how oxytocin impacts on female maternal behavior and vasopressin for male pair bonding and parental or paternal behavior.
Now, here's what I don't know: is it monogamy that produces the higher levels of oxytocin, or is it higher levels of oxytocin that produce the monogamy?
Well, for oxytocin is primarily involved with the maternal behavior. It's vasopressin that's associated with monogamy. And what's quite interesting is some of the same teams, so Insel and Young, who worked on how vasopressin relates to pair bonding and paternal behavior in voles, have also shown that you can take vole species that are not monogamous and add vasopressin and make them switch to being monogamous. So you can actually change the hormones, and that totally changes their behavior. In a larger context, one of the questions that drives our research is understanding what makes humans special, or what is it about human biology that is both similar and different to other primates. And one of the things that's very remarkable about humans is that we have a very large brain and we have exceptionally long juvenile and infancy periods. And because we're born so helpless and so unable to take care of ourselves, parenting becomes exceptionally important. And human babies are pretty much totally defenseless, and even throughout their juvenile period, they really need much more investment by their parents than similar species that are closely related to us. And I think that tells us that in humans particularly, understanding the role of parenting behavior and the biology of parenting behavior is very important.
Chuck, let me turn to what's the effect on children who are deprived of nurturing.
We know that children are exposed to a continuum of care. And as you can see here, this infant and this mother are having a wonderful conversation. You can see the mother's clearly in love with this baby. And this is what we want to see in all infants and mothers. What happens, though, is under conditions of profound deprivation, which we'll see in just a moment, all of that is missing. So here we have a pair of twins actually interacting. But now we have institutional care. And a couple of things to note is the sheer number of babies that are sitting in these institutions, the lack of caregivers. You can, as you look through here, there's one you can see in the back. So you can see that there's a very low investment in these children, unlike the video clips we saw in the beginning. And this lack of social interaction, we think, plays a fundamental role in building the brain. So what we started to observe was, in this study, what happens to the developing brain in kids growing up in institutional care. And we actually performed a manipulation in which we saw a large number of children abandoned to institutions in Romania. And after studying them extensively, some of them were then placed in very high-quality foster care, and some remained in the institution. I want to show you a video of what a child at around the age of 2 looks like. So this is on an outing with a bunch of little kids from an institution. And if you notice the girl who's rolling over, she's 22 months of age. Her IQ is below 50 at this point in time. She's been in the institution close to the time of birth. Notice the other little girl is rocking. And off camera, there's 3 or 4 other kids, all of whom are rocking. And that stereotype rocking is very characteristic of kids who grow up in institutions. So the question then becomes, what happens to the brain? So in the next slide, I'm going to show you beginning of this journey to understand the brain. The first thing we did is we recorded the brain's electrical activity, or the EEG, and we do that by simply placing sensors on top of the head. The billions of neurons we have in the brain generate electrical activity that we can pick up, and we can then, from that electrical activity, basically infer the power that the brain is producing, how much electrical activity is there. We can color code that indicates more or less power. So on the right side, you see an image of a never institutionalized brain. And you're looking at this from the top down, and the distribution of electrical activity is portrayed here to reflect much more brain activity in red, sitting really over the frontal lobe. But if you look on the left panel, that's the institutionalized group. And you can see that the brain of the kids in the institution are basically underpowered. So we can use the analogy, instead of a 100-watt light bulb, it's a 40-watt light bulb. At that point, we became extraordinarily concerned and thought, what's going on? What's making the brain produce less electrical activity? So when the children were 8 to 10, we performed magnetic resonance imaging on them to look at the detailed anatomy of the brain. On the right is an MRI scan, and we're showing here gray matter. Gray matter really represents the cell bodies and appendages of neurons that sit on the cortical surface and really do the computations and the calculations that the brain does. Whereas white matter, which I'll show you in a moment, really does the communication of the brain. And white matter shows up whitish and gray matter shows up grayish. So if you look here, on the far right, you see the amount of gray matter in the never institutionalized kids. These are the children who grew up in families in Bucharest, Romania. But if you look at the children who've been institutionalized on the far left, it's dramatically reduced. They show less gray matter, as do the kids we put in foster care, and I'll come back to that. So what this is showing us is that the brain has much less gray matter by a function of being in an institution. So in the next slide, we show the same reduction in white matter, this communication pathway. What's scary about this is it's almost suggesting that we know that there's a smaller brain as a function of being in an institution.
The reason this is so important is because before Chuck did these studies, people knew that deprivation or lack of parental interaction was bad for the cognitive development of the children. One didn't know to what degree it affected the brain directly. One thought this was likely to be the case, but this was the first evidence that showed dramatic changes occurring in the brain.
Would it be different if somehow there was some kind of activity with the kids who were in institutions? In other words, there was a much more collegiality in some way that was produced from outside?
Yes.
So in a moment, we'll talk about what happens when you put kids in families. Your question really is, can you improve an institution? And
Charlie's asking another question. Does it have to be the parents? Could they be parental substitutes?
Right. So we know that it has to be the investment the caregivers make. It doesn't have to be the parents, but it's got to be caregivers who care about that child. And what goes on in institutions is you don't have that investment.
And is it as simple as you can feel the sense of your brain will develop if you can feel that there's some contact, that somebody knows who you are and recognizes you, and there's some act of caring
affection
for you.
Yes.
Affection.
Yes.
Precisely. And as Eric said, I think it's this social interaction that really is what's stimulating brain development. And it's the lack of social interaction. So kids in an institution who got cognitive stimulation and linguistic stimulation, but no caregiving, they'd be just as poorly off as the kids that we see. Now the question is, how much recovery is there? Right. So in this study, we placed half the kids into foster care, really high-quality foster care. In the beginning of this video, once again, we'll see that little girl, when she's 22 months of age, IQ below 50, in an institution. Very atypical motor behavior. Right after this, we put her in foster care, and this is now her in foster care 8 months later. Her IQ is now in the mid-60s. This is her interacting with her foster care mother. It's clearly they have this loving relationship, and this is merely 8 months of foster care. And in this clip, she's now about 4 years of age. She's now spent half her life in foster care. And look at that interaction she's having. This is the same little girl we saw in the beginning who was crawling backwards, rolling over, had barely no language, and now her IQ is in the 80s. So there's tremendous recovery that can occur by placing children into a family, but it seems to be regulated by a critical period, by which we mean placement before the age of around 2 years of age leads to much better outcomes, but placement after 2 years of age leads to much less desirable outcomes. So on the next slide, we can show the EEG. Again, to remind us, on the right is the brain of the never institutionalized child. More red means more activity. Left side is the institutionalized child. In the next slide, we see the critical period. So now the brain that says left at institution after age 2, notice it looks identical to the institutionalized brain. Child's in a family, but not until they were older than 2. In the next slide, you can see that now the children placed before 2 have brain activity that looks just like the kids who've never been in an institution. And we see this in other domains as well. So what we see then is this inflection point in development. Removal from an institution and placement into a good family before 2 leads to much better outcomes than children who are placed after 2.
So timing is everything, and if you don't have it, it puts a certain ceiling on how much
exactly
you can produce.
It's as though the window of opportunity for brain plasticity is reduced.
What we can bring to bear on this subject is not only a deep discussion of what the sort of psychological and sociological issues are, but what the biological underpinnings of that is. When we begin to speak about the biological underpinnings, we want to distinguish, as you already implied, 2 different concepts: anatomical sex and gender identity. Anatomical sex is the body parts associated with sexuality and reproduction. Gender identity is a more subtle, complex image that is the sense of oneself as a male, female, or something else. Let's begin with the anatomical sex. Anatomical sex is determined, not surprisingly, by our genes. Genes are arranged in chromosomes, and we have 23 pairs of chromosomes. We get half from our father, we get 1 pair from our father and 1 pair from our mother. 22 of these are called autosomes, and the difference between the father's and mother's contributions are real, but they're modest. But the sex chromosomes, the difference is really quite profound. Women are XX and men are XY. The chromosomes are really quite different than the autosomes, and they have a very important function in determining sex. Let's begin with the Y chromosome and see how that determines the sex of the male. We are born with an undifferentiated gonad that can move in either direction. It either can develop into testes or into ovaries. If there is a Y chromosome there, it has a region in it, which is called the sex-determining region of Y, that contains a gene that activates the differentiation of the undifferentiated gonad into the testes. If that Y gene is not there, you have an XX, you have a female gonad develop. And each of those has profound consequences. Let's look at the male differentiation first, the male testis. The testis develops within the first 7 weeks in utero, and if the testis matures as a result of the sex-determining region of Y, the testis releases a massive amount of testosterone, comparable to the level that you have at puberty in the adult. And that is responsible for giving you the male body form the brain characteristic of male functioning, as well as having actions on sort of every aspect of your being. If the sex determining region of Y is not there, if you have XX, you have development of the ovary. The ovary, in turn, secretes estrogen and progesterone, and it gives you the female body form, and it gives you changes in the brain. So clearly, these are extremely important changes, and that's the easy part. This is the anatomical part. The more subtle, the more elaborate part is the gender identity.
Let me begin with Ben. Tell me the experience that you went through. Share with us as much as you can about what led up to your decision, how you carried forward, and the impact it's had on your life.
Sure. I think in many ways my experience is probably typical for other transgendered people. I was, I think, only about 4 or 5 years old when I first started to have strong feelings that I felt more like a boy. I was born as a girl, Barbara, but I felt like a boy. I played with little boys. I preferred boys' toys. I remember dreadfully wishing I could be in the Cub Scouts and Boy Scouts. Every Halloween I would dress up as an army man or as a football player. This all just seemed very normal to me. I felt like a boy. But as I got into middle school, early teen years, I did start to feel more and more uncomfortable with this. I didn't feel I should have breasts. I didn't feel comfortable at all wearing dresses, makeup, jewelry. And it became increasingly uncomfortable when I got into high school. I started to be teased more by kids. And, I just had a lot of confusion about my gender, and I felt very ashamed of it. I never spoke with friends or family about it at all, ever once.
No one?
No. I just felt very ashamed and very confused. Don't forget, this is the days before the internet, so there wasn't a lot of information about this sort of thing. So as I got into my 20s, I was doing well. Although I didn't feel good about my gender, I was doing well in my career. I was doing medical training and research training. But I was increasingly uncomfortable, and like many trans people, I started to think about suicide. I never actually attempted suicide, but I thought about it a lot. And this is actually a picture of me back when I was Barbara. I think I was 30 at that point, and I was a bridesmaid at my little sister's wedding. And I can still remember vividly, even though that was 30 years ago, just the agony that I felt, the discomfort putting on that dress, wearing jewelry, wearing makeup, and still 30 years since I still remember that. But anyway, I did complete my training and begin a job at Stanford about 20 years ago when I was age 40. And about 2 years into that, I actually developed breast cancer. And I was still very confused about my gender identity, but I knew that I didn't like to have breasts. And so when the doctor said he needed to do a mastectomy to remove the cancer, which very fortunately was picked up early and I was cured of, I said, while you're there, please take off the other breast. And he was quite horrified by this. Perhaps he was the first one I shared my feelings about gender identity, but since this cancer runs in my family, he did agree to remove the other breast. And I just can't tell you how therapeutic that was. I felt so relieved to have those breasts removed. And I recognized that was a very different response than my mom had when she had her mastectomy. She saw this as a huge blow to her femininity. But after the surgery, the doctor started talking to me about reconstructing the breasts, and I was absolutely horrified. I said, there's no way you're putting those things back on me. So this was sort of increasingly a clue to me that really there was something a little bit different about my gender. And it was only about a year later, I was reading the San Francisco Chronicle one day when I read a several-page article about the life of James Green, an openly female-to-male transgender person in the Bay Area. And I realized for the first time in my life that there were other people that had experienced the same sorts of gender confusion, that there were other people like me and that I might be transgender. And so I went to see Don Laub, who was a sex change pioneer at Stanford, and he ran a gender clinic. And indeed, after they evaluated me, they told me that I thought I was transgender, and they offered me the possibility of changing my sex, which was immediately irresistible to me. And very quickly, within weeks, I decided to change sex. I'd already had the upper surgery, the mastectomies. I did not want lower surgery, but so all that was needed was to take some testosterone, and you can see the effects that it's had on me. It's powerful stuff. And one of the most surprising things about the testosterone was that I actually, to my great surprise, it became much harder for me to cry. And male to females report that it now becomes much easier for them to cry. So that was perhaps a surprising experience. But I think the main experience that I had is I just felt after I changed sex, it's hard to describe the intense relief that I felt, just like this weight lifted off of my back, and I've been so much happier since. I've never had another suicidal thought. I have to say that at the time I decided to do this, had been a scientist at Stanford for several years, I was very worried that changing sex would harm my career. This was 20 years ago. But I have to say that everybody, all my colleagues and friends and family were immediately supportive, and I've been very fortunate to have my career continue and to have lots of wonderful students and so forth.
I want to show you a very powerful example of identical twins who, in this case, one of the male twins, and we have proven that these twins are indeed absolutely identical and born male, but one of them at age 3 started to say everything about being a girl. And in fact, if there was any issue that came up, she turned it into an issue of gender. And she did some of those things that we consider hallmarks, like preferring to wear female underwear and female pajamas, and etc. And by the age of 7, the family decided, with the help of a counselor, to change her name to a female name and to have the child assume a female role. So here is twin sister with twin brother at the age of just about 9 to 10 years of age.
4th grade.
And so they were 4th grade. And what I want to point out is that if I switched all the attachments to them, the earrings, the clothing, the hairstyle, the shoes, you could basically switch one for the other. And the fact of the matter is that kids with their clothes on are virtually interchangeable pre-pubertally. And actually, their hormonal levels are virtually, at that particular point, interchangeable. So everything happens really at puberty. So here they are at age 14. Now, because it is so difficult to live in a gender different from your biologic sex, when you have the toxic effects of your genetically hormone-driven puberty, which would basically make twin sister look exactly like twin brother. And you can see that she looks still almost like a 9 or 10-year-old, and there's a good reason for it. She's had her puberty suppressed. The next slide shows the level of sex hormones across the human lifespan. Look at the blue line, which is the male level of testosterone, the hormone that Ben was receiving and that some of us make, but that during fetal life, especially in the mid-trimester, the level of testosterone in a fetus rises to a level close to the full adult range. And then it falls, and then there's another blip up right after birth, a kind of second puberty, and then things go completely dormant. In fact, if they didn't go dormant, we'd have a whole bunch of pubertal-looking 5th graders running around because everything is suppressed.
When we're thinking about possible causes for transgender, and Ben and I spoke about this before, wouldn't it be possible that some aberration in testosterone secretion, for example, or estrogen secretion during intrauterine development or shortly thereafter might be one of the contributing factors?
It's certainly possible. It's certainly possible. It's a very dynamic stage. We still don't know what that second bump is right after birth, or what role it plays, or whether boys who are born without testes but are otherwise normal show any differences as a result of that. The problem, Eric, is that when we want to look at a hormone, we have to be able to measure it. We have to get it out and measure it. Well, you can't get it out of the brain. The other problem is sometimes it isn't the hormone level that's important, but the affinity of the receptor for the hormone. And it's very, very difficult to measure such things.
This is really defining a really fascinating biological problem, to see what is the biological underpinning of transgender identity.
Exactly.
Because, as you point out, it's so important because kids who are in the wrong sex, their incidence of suicide attempt is very serious.
Oh, it's one of the highest risks of any.
Yeah.
So,
this is really something we need to understand.
In the pubertal process, this whole system reawakens again. That system that had been awakened in utero suddenly comes back, in which hypothalamic hormones hitting the pituitary cause the release of other hormones that strike the ovary or the testis and cause the release of what we call the sex steroid hormones, which are testosterone and estrogen mainly. And those things produce the differences between the body of the male and the female associated with puberty. So we have been able to, probably since the 1980s, we've been able to block the release of the hormone from the hypothalamus to the pituitary. And once you do that so far upstream, everything downstream goes down to zero. And we have a record of this medication being completely successful in shutting them down until an appropriate time, and also the fact that it's completely reversible. Look how revealing this is, because the twin sister affirms a female identity and her puberty is blocked for 2 years. 2 years to get more time for counseling without the pressure of body change. That's very important, because if we're going to give her estrogen, that's going to have permanent effects. So take a look what would happen to her if she hadn't been given this blockade. She would look exactly like twin brother. And he's in early puberty for a 14-year-old. But so would she have been, because they're identical. Now, here are the twins at age 17. At age 14, just after that picture was taken, she began estrogen, while at the same time having her male hormones blocked. With that, our patients don't need breast surgery when they feminize. Because it is so effective. And she is now, in that picture, entering junior high school, as is her brother, and she's absolutely fabulous. This past October, the Dutch, who taught us this, reported the first follow-up of the patients, 55 of whose puberty was blocked, and for whom sex steroids were later switched in the manner of our twin sister. The Dutch group all had surgery at 18, at which point their gender dysphoria, their total uncomfortableness with their gender, disappeared. And the Dutch found that the kids treated this way as psychosocially functioning as well or better than the control group of non-transgender kids they were being compared to.
Is that a powerful argument for surgery?
It's a powerful argument for available surgery at the right time for the right people.
But also, I think one of the nice things about the Dutch approach is that it delays puberty. Once puberty sets in, you have, as was just pointed out, physical changes that make it much more difficult to reverse. So you prevent those physical changes of the other sex occurring. Moreover, in addition to the biological changes that it delays, it allows the child to think through whether this is the course they want to be on. Because some people say, I'm in the wrong body, I want to be a man when they're a woman, but change their mind after a few months. Think this is the wrong course. That occasionally happens. This delay allows not only the physiological process to be handled in a more satisfactory way, but the psychological evaluation to see whether this is the way the kid actually feels, whether it's only a temporary decision in their minds.
So the Dutch gave me their protocol in 2006, and we started using it in Boston Children's Hospital, at which time we were the only major medical center to do so. But we're not that many years since 2006, and over 40 programs now are doing
right here in the Times article.
So it's now becoming the standard of care.
For more about this program and early episodes, visit us online at pbs.org and charlierose.com.